Hi everyone, my name's Olivia, and I decided to do my project on multiple sclerosis and a possible treatment stemming from human placental cells. Just to briefly review multiple sclerosis and its pathology again, we know MS as a neuroinflammatory disease affecting the central nervous system, and it's autoimmune in nature because of the autoreactive lymphocytes, or T cells and B cells, that modulate the inflammatory response in the CNS. These lymphocytes enter into the blood-brain barrier. They release pro-inflammatory cytokines, which go on to induce this massive inflammatory response, which induces death of the oligodendrocytes, myelin damage, and it also creates glial reactions. And it's also believed that B cells are implemented in making antibodies against myelin, but this mechanism hasn't been fully studied yet, but it is postulated to be involved in MS. The demyelination that characterizes the disease is what also produces the characteristic plaques of MS. I don't know if you guys can see my cursor, but here in the MRI on the right, the MRI scans on the right, you can see the white spots. Those are the plaques that characterize MS, and you can see between the initial and the 18-month scans, there are more plaques developing in the occipital and temporal lobes. There are 4 different disease courses of MS. Relapse remitting is the most mild, primary progressive, secondary progressive, and then primary relapsing is the most severe. And in MS, we do see such a large variability in pathology because these plaques can develop pretty much anywhere in the CNS, which is going to give you variable symptoms as well, which is why MS is so hard to diagnose. But the most common symptoms that we do see are sensory disturbances, cognitive deficits, affected vision, muscle weakness, as well as bowel dysfunction. Oh, as well as pathology is going to be affected by the severity and the number of lesions that you see. Unfortunately, as of right now, no current treatment actually cures a patient of MS, but it's really exciting to see that since 2017, we've had 3 new immunomodulatory therapies approved by the FDA. Mayzent works to basically acts as an anti-inflammatory by modulating lymphocyte release from lymph nodes as well as limiting blood-brain barrier penetration of these lymphocytes. Mavenclad, which is actually previously approved to treat hairy cell leukemia, is now approved to treat relapsing remitting MS. It basically depletes T cell and B cell count through various cellular mechanisms. There's a lot, so I don't really have time to talk about all of them, but Ocrevus also exerts anti-inflammatory effects by depleting the number of eligible B cells to cause inflammation by targeting the CD20 antigen present on certain B cells. It basically just inactivates the B cells, Ocrevus binds, and the B cell is no longer able to work with the T cell to induce a strong inflammatory effect. And that mechanism you can kind of see here in the photos on the left. So the proposed treatment that I decided to look at is called PDA1. It's previously known as Simplacell L. It's an immunomodulatory therapy utilizing cells derived from a full-term human placenta. And PDA1 had previously exhibited reductive properties in disease severity and disease progression in a human MS mouse model experiment without any signs of paradoxical disease worsening. So this study, this phase 1 clinical safety study, was merited to confirm a lack of disease worsening in human patients before PDA1 went on to further clinical trials. This study utilized 16 patients with either relapsing remitting or secondary progressive MS. 8 patients were assigned to the low-dose group, receiving 1 injection at the specified concentration you see on the screen. The 8 remaining patients were allotted to the high-dose group, receiving 4 injections at that specified concentration of cells. And 2 patients from the low-dose and 2 patients from the high-dose groups received placebo injections. All injections were given on day 0 and day 7 of an 8-day treatment phase, which followed a 35-day pre-treatment screening phase where patients underwent baseline MRI scans to obtain baseline lesion numbers, as well as a baseline expanded disability status scale scoring. Following the treatment phase, patients were evaluated for 12 months, and the first 6 months of this evaluation phase were allotted simply for safety and efficacy analysis. So basically just to see if there was any paradoxical disease worsening present. Patients were required to undergo monthly MRI scans, and this was to compare if there were any new lesions present in these monthly MRI scans after treatment. They could compare it to their baseline number of lesions obtained during the pretreatment screening phase. The authors defined paradoxical disease worsening using what's called Cutter's rule, and it's basically the formation of 5 or more new gadolinium-enhancing lesions on 2 consecutive monthly MRI scans. What is a gadolinium-enhancing lesion? So gadolinium is actually an earth metal, or an earth element, that's used to enhance MRI contrast. It's put into a contrasting agent, and if a gadolinium-enhancing lesion is present, you will see a very stark contrast between the lesion and the unaffected tissue. And this lesion often signals a breach in the blood-brain barrier and heightened inflammation as a result, because the contrasting agent and subsequent immune cells, like lymphocytes, are better able to penetrate the blood-brain barrier into the CNS and induce an inflammatory response and more demyelination. So we can see here in figure 2 that there was one patient in the back left corner that did have an increase in the number of gadolinium-enhancing lesions present, but because the flare happened at 5 months after the injection, this did not meet Cutter's rule. But no other patient actually met Cutter's rule, which is great and shows really promising results. So for the next 6 months of this evaluation period, this was called the intent to treat phase, and basically the authors wanted to see if PDA1 were intended as a treatment. In this case, did it show any efficacious results in improving a patient's quality of life or the severity of their disease? The changes in disability were quantified using the Expanded Disability Status Scale, or EDSS. In this case, a higher score signifies greater disability. And as you can see, actually 13 out of 16 patients showed a similar EDSS score or a decreased EDSS score compared to their baseline report, which is really promising. And the authors took this as a possible reparative component of PDA1 in relapse remitting and secondary progressive MS. There were several patients who reported low-grade adverse infusion-related events such as site swelling and hematoma in response to the injections, but these did not impact any quality of life, and they resolved themselves rather quickly. There were 2 patients who reported serious infusion-related events, like an anaphylactoid reaction and a superficial blood clot, but these also resolved themselves without any changes to treatment, which is really promising because these patients did not have to be excluded from the study, and there were no serious changes to quality of life. But overall, I think that this study really shows how well tolerated these PDA1 injections were, and also that they have a potentially efficacious role as a therapeutic in treating relapsing remitting MS and secondary progressive MS. Not only is the source of the PDA1 placental cells traceable, but it's also safe, coming from a human source. It's also going to be in ample supply as human reproduction is constantly occurring. And even though this study did have a relatively small sample size, it did merit a proof of concept study that's going to further explore the dosing of PDA1 in treating relapsing remitting and secondary progressive MS. I'm anxious to see how this treatment pans out in the future, but in the meantime, thanks for listening and good luck with finals.